inside of a bacterial cell is folded. You can see right there it's folded. And if you can break down it, it's a very, very long. Somehow about 1,400 micrometers. This is 200 to 700 times of the size of bacteria. And I tell you one thing, how many gene base pairs there? 0.5 to 10 million gene base pair. How many genes? 500 to 10,000 genes there. Okay, you need to know the difference between base pair and the genes, is that right? It's not all the genes, base pairs are genes. The gene, which means the sequence carry on genetic information. If you forgot, go back to see your high school biology textbook. Okay, what means what means the genes? For E. coli, we talk about E. coli all the time. That is about 4.6 million base pair. Okay, that's for the chromosome. Then, very interesting, the chromosome most of the time is circular. And there's some linear chromosome. And in the chromosome, they could have something which are the very small sequences. They could be added there. That is called a plasmid. So the plasmids most of the time is linear or circular DNA sequence. And they could be integrated into a chromosome. If it's integrated, we gave them a very special name. In endosome, we think right here we have its, if it's integrated, let me see, did I find the name here? Episome, sorry. If it's integrated, we call it episome. And the plasmids, they could also be isolated, and they also could be do replication, which is independent. The plasmids, the gene base, most of the time is around 30 to 100 base pair. But I want to tell you one thing, some of them, it's carrying on very important information. For example, the all plasmid, it is basically responsible for antibiotic resistance. That's called all plasmid. Okay? Plasmid, I mentioned, could be circular, could be linear. It really depends on different bacteria. So when we know Borrelia burgdorferi, they have 12 linear and 13 circular plasmids. So it really depends on the different bacterial construction. Okay, the last thing. I haven't been talking about this, but I want to give you some of the hints. What is DNA? You heard about this all the time, is that right? It's too simple. I mean, what is DNA? 2' deoxynucleotide. So what's the structure? Did you ever thought about that? What's the structure of DNA? Or what's the structure of RNA? What looks like? 5 carbon sugar, is that right? Right here. That's called the ribose. Carbon number 2. You have the OH right here, hydroxy bonds. If this O is gone, that is DNA. If the O is there, it is RNA. Now what are the others? Phosphates. Is that right? You need a phosphate. How about the top here? A, T, G, C. That's the gene base. So what is the DNA structure? Phosphate, 5 carbon ribose, and the gene base. So I always talk about that. I said the DNA is a very boring chemical because it's so simple. It's very boring. You have a phosphate, you have a 5 carbon ribose, and then you have a gene base. That's it. But protein is more complicated. Primary, secondary, tertiary structure, quaternary structure, and the peptides, the enfolded alpha, helix, C, beta sheets. Is that right? It's more complicated. The sequence is difficult to do. The DNA is easy to do. And DNA will be composed by the plasmids. Is that right? There will be 30 or 100 BP base pair based on the gene complementary theory. And this is T, this is A, this is 300 bonds C, and this is 300 bonds G. Okay? So we'll talk about this again. In exam 3, this is a very important information there. We're also going to talk about the history about that, because there are five very interesting, very important study we could talk about. Okay? Okay, that's all we have today. And we'll continue to talk on next Tuesday.
I'm big on that.